Now we'll start with Kingdom Protista. And now under Kingdom Protista, all the organisms are single-celled eukaryotes. What are these? Single cell in the sense unicellular eukaryotes. There are again some subgroups under Kingdom Protista, namely chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans. Which organisms were included there? Bacteria. Those are prokaryotes. Here you have unicellular eukaryotes. And remember the groups here. So we are going to see each of these groups in detail. Chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans. Now these are primarily aquatic, which means mainly in water. Now, what are eukaryotes? Eukaryotes will have a well-defined nucleus and other membrane-bound organelle, because this is not seen in the prokaryotes, right? In prokaryotes, you don't have a well-defined nucleus. You just have something like a nucleolus, which is not going to be well-defined. It is just going to be floating in the cytoplasm, right? But in eukaryotes, you're going to be having a well-defined nucleus and other membrane-bound organelles. So if this is, let us say, drawing a rough one here, let us say this is a mitochondria, it's going to have like a membrane around it, right? So these are membrane bound organelles, which you do not see in the prokaryotes. It's seen in eukaryotes. Now, some of these are going to have flagella or cilia, which is important for movement. They will reproduce asexually and sexually by a process involving cell fusion and zygote formation. So both kinds of reproduction is possible here, asexually or sexually. If it is sexually, then you have cell fusion, right? So you have the gametes fusing, right? And then when the gametes fuse, you're going to have a zygote formation. Now let us start with the first subgroup of Kingdom Protista, which is the chrysophytes. This group, okay, includes diatoms and golden algae, which are also called as desmids. So diatoms and golden algae are the members of chrysophytes. They are found in fresh as well as marine environments. So you can have your fresh water as well as marine water, which is nothing but salt water, right? So fresh and marine environments. They are microscopic, that is, you cannot see them with your naked eyes. They will float passively in water currents, also called as planktons. What do we mean by planktons? In the sense they cannot move on their own, but whichever way the water moves, whichever way the water current takes it, it will just passively flow along with that. So such organisms are called as planktons. And most of these are photosynthetic, which means they are making their own food. So please make a note of this. Now what you see on top over here, this is diatom as seen under the microscope, and down here this is a golden algae again seen under the microscope. So here please make sure that under Kingdom Protista you are writing this chrysophytes. I want you to take down certain points, at least write down this point. Diatoms, golden algae, write that they are planktons. Now this plankton word you should know irrespective of whether it is part of chrysophytes or no. What are planktons? Organisms which are floating passively along with the water currents. These are planktons. Now the diatoms here, they have cell walls which form two overlapping shells which fit together, something like a soap box appearance. So these are the two shells of the diatoms, and they are very thin. The cell walls, okay, these are going to be forming like overlapping shells which will fit together, just like how you would have seen a soap box. Similarly, now the walls are embedded with silica and the walls are indestructible. Thus, diatoms have left behind large amount of cell wall deposits in their habitat. So, even once they die, what happens is the cell wall deposits are still going to be there because the walls are indestructible and they're embedded with silica. Now, this accumulation over billions of years is called as diatomaceous earth. For diatoms, cell walls form two thin overlapping shells which fit together, resembling a soap box. What are the walls made of? They are made up of silica. And accumulation of these diatoms over the years is called as diatomaceous earth. Now, being gritty, this soil, this diatomaceous earth basically, is used for polishing, filtration of oils and syrups. So it's used as a filter. You filter anything through it, what happens is all the dirt or whatever that is there, it'll get trapped in the diatomaceous earth, and the filtrate that you get is going to be very clean. So that is for filtration of oils and syrups. Now, these are the chief producers in the ocean. Here we said these are mostly photosynthetic, right? So these are the chief producers in the ocean. This is about diatoms. And the next subgroup is the dinoflagellates. These are mostly marine and photosynthetic. Here, when we saw the chrysophytes, what did we say? They are found in fresh as well as marine. Here, dinoflagellates is going to be mostly marine and photosynthetic. Now, these can be of different colors. These organisms, they appear yellow, green, brown, blue, or red, depending on the pigment which is present in their cell. So, if you have a yellow color pigment, it's going to look yellow. So, depending on that, it can be of these 5 types of or colored organisms. So, here, what was the cell wall made up of in diatoms? It is silica. Okay, in dinoflagellates, it has a stiff cellulose plates rather, as you can see over here. Okay, these are all plates that you see, right? It's made up of cellulose. Now, most of them are going to have two flagella. The two flagella, one lies longitudinally and the other lies transversely in a furrow between the wall plates. What do I mean by this? You see in this diagram here, longitudinal flagella, this one and this one. Longitudinal is the one that runs from up to down, okay, lengthwise. Then here, if you see, there's a transverse flagellum which runs from left to right or side to side. So even over here, if you see, this one would be the longitudinal flagella, and this one that runs side to side would be the transverse flagella. So please make sure you're writing this about the dinoflagellates, about their cell wall. I think pigments you will be able to remember, or you can just note it down: yellow, green, brown, blue, red. Cell wall, and the main thing over here is their flagella. One is longitudinal, the other is transverse, and it is between the plates. Now, we have something called as the red dinoflagellates. Remember, here there are many pigments. Right? So the red dinoflagellates, okay, the scientific name is goniolax. What do is they rapidly multiply in the sea and they make the sea look red, and it's also called as red tides. So what you see here, okay, this is nothing but red tide. So when does this occur? When the red dinoflagellates or the goniolax rapidly multiply such that they multiply all over the sea and they give this red color appearance. The toxins released by such large number of dinoflagellates may kill other marine animals such as fish. So what happens is sometimes since they are growing in such a large number, the toxins released also are going to be large. This will pose a threat to the other marine animals which are present, right? The fish and other maybe the plants. So their toxins can be harmful there. Now, the third group here is the euglenoids. Okay, now majority of these are freshwater organisms which are found in stagnant water. So that is stagnant water. So that is where you will usually find the euglenoids. Again, over here, this is freshwater stagnant. Now, instead of a cell wall, these organisms have a protein-rich layer called as the pellicle, which makes their body flexible. They do not have a cell wall. Instead, there's a layer called as the pellicle. This pellicle is protein rich, made of proteins. They have two flagella. Again, over here, you're encountering two flagella here. But see the difference. You're going to have one long one and a short one. So here, if I have to show it to you, this is the long flagella. And then you have the second smaller flagella over here. But what did we see in dinoflagellates? There the flagella, one is longitudinal, one is transverse. Here you have two, but one is long, one is short. That's the difference. Now, the euglenoids, in the presence of sunlight, they can carry out photosynthesis, make their own food. But in the absence of sunlight, they act like heterotrophs. Otherwise, they act like autotrophs. So in the presence of sunlight, they act as autotrophs. But in the absence of sunlight, okay, they act as heterotrophs, wherein they will predate on other smaller organisms. So obviously, without sunlight, they cannot carry out photosynthesis. So they will predate on other organisms for their food. Now, the pigments of the euglenoids are identical to those that you see in higher plants. You have chlorophyll A, B, okay, xanthophylls. Those are the kind of pigments that you see in euglenoids as well. An example is euglena. So what you see here, euglena, which is a euglenoid. Stagnant water, protein-rich layer pellicle, two flagella again here. And you should know, presence of sunlight, they are photosynthetic. Absence of sunlight, they are heterotrophs. And this example, euglena is an example of euglenoids. The fourth group is slime molds. These are saprophytic protists. The body moves along decaying twigs and leaves engulfing organic matter. So I'll show you a video after this wherein they actually show how the slime mold moves. Okay, so as it moves, it is going to engulf all the organic matter. It could be twigs, it could be leaves, okay, any dead material, anything. Now there are two what do we say, situations that we can see here. One is under favorable conditions, the other is under unfavorable conditions. Now under favorable conditions, they form an aggregation called plasmodium, which may grow and spread over several feet. Aggregation in the sense like a collection. So they will all aggregate together. Okay, this aggregation is called as plasmodium, and this will grow and it will start spreading over a big area. This is under favorable condition. If there is unfavorable condition in the environment, then the plasmodium will differentiate, as in break apart, okay, and they will form fruiting bodies bearing spores at their tips. So something like this. There will be fruiting bodies which is going to have spores at the tips. So this is during the unfavorable conditions. Okay, so you should know this. How do they behave under favorable conditions? How do they behave under unfavorable conditions? Now the spores possess true walls. The spores are extremely resistant. They will survive for many years and they are dispersed by air currents. So why do they need to survive for many years? Because maybe in that particular environment, the unfavorable conditions are for many years. Once the conditions become favorable, they will again germinate and they can again form the plasmodium aggregation. So over here in the picture that you see, the yellow thing that you see, right, that is a slime mold. There also, malaria is caused by a parasite called as plasmodium, but that is not the same as this one. These are two different entities.